My name is Jim Haber. I'm a professor of biology at Brandeis University in Massachusetts and a member of the steering committee of the Coalition for the Life Sciences, who has arranged the speakers list for the 21st season of the Congressional Biomedical Research Caucus. I'd like to thank, first of all, the chairs of the caucus, Representatives Brian Bilbray, Charlie Dent, Jackie Spear, and Rush Holt for their commitment and dedication to the support of this caucus, and without them, these very timely presentations really couldn't happen. And also I should thank the Howard Hughes Medical Institute for the support of the caucus through a very generous grant, and obviously we are tremendously grateful for their support. I should tell you that all of these presentations are videotaped, and that you can go to the Coalition for the Life Sciences website and you can see them again and also register for RSS feed so you will be alerted to any future postings. So it's my great pleasure to introduce Dr. Mark Baer from the Massachusetts Institute of Technology. Dr. Baer is an investigator of the Howard Hughes Medical Institute and Pickauer Professor of Neuroscience in the Pickauer Institute for Learning and Memory, and also the Department of Brain and Cognitive Sciences at MIT. He came to MIT in 2003 after spending 17 years on the faculty of Brown University. He has his Bachelor of Science degree from Duke University and his PhD in neurobiology at Brown. He then did postdoctoral work in Frankfurt, Germany, and returned to Brown University on the faculty. Among his many honors, he is elected as a Fellow of the American Association for the Advancement of Science, the American Academy of Arts and Sciences, and the American College of Neuropsychopharmacology. In 2006, he was the recipient of the William and Enid Rosen Research Award for outstanding contributions to our understanding of Fragile X by the National Fragile X Foundation, and it's my great pleasure to introduce him.
Thank you. Thank you very much, and good afternoon. So let me begin with the good news. So this year, there are now mechanism-based treatments in human clinical trials for the treatment of Fragile X syndrome, which is a leading known inherited cause of autism. And I think I was invited to come down and brief you today to sort of give you an idea of how we got to this point. We don't know the outcome, of course, but we're extremely hopeful. But how do we get to this place? And I want to begin by making a confession that may surprise you. And the confession is that I am not an autism researcher. I am a basic neuroscientist, a basic neurobiologist, and you'll see, hopefully in a few minutes, how it is I've been drawn into the world of autism research. But just so we're on the same page, what is autism? Probably don't have to explain this to any of you anymore because it's a very prevalent condition. But it's characterized by three major symptom groups. One is reduced social behavior, abnormal language or lack of language, and repetitive and restricted behaviors. In addition to these core features of autism, most individuals with autism have comorbid conditions, the most prevalent of which is intellectual impairment. About 70% of those with an autism diagnosis are also intellectually disabled. A very large percentage also suffers from seizures, particularly during early childhood, about a quarter of those with the autism diagnosis. And other features, this is not a complete list, include altered gastrointestinal function, sleep disturbances, and so on. Now, autism is highly heterogeneous, which is to say that it presents differently in different individuals. And so really, these days, no one expects autism to be a one entity. Rather, there are many causes of autism. Autism seems to be a final consequence of many different injuries or insults to the nervous system. And so today we talk about an autism spectrum and whether individuals fall on an autism spectrum with varying degrees of severity of the symptoms. As I mentioned, it's highly prevalent. Now the estimates are that almost 1% of newborn babies in the United States this year will have a diagnosis of an autism spectrum disorder by the time they turn 3 years of age. So it's a serious human health concern. It's clearly a disorder of brain development. I think that should go without saying. What we do know about autism is that one of the features of brain development that seems to go awry is communication between neurons at the specialized sites of communication called synapses. And I'll tell you a little bit about altered synaptic function in autism. And finally, autism is a highly genetic condition. That is to say, if an individual is born with autism and has an identical twin, there's a 90% probability that twin will also develop autism. So there's a clearly very high genetic risk factors in autism. And in a way, it sets it apart from many of the other psychiatric disorders, which are also highly genetic, but probably not as high as autism. And we now live in what's called the post-genomic era, where we've now sequenced the human genome, and with the promise that that understanding would lead to the discovery of the genetic alterations that confer risk or cause disease or confer protection from disease. And this is the same in autism and psychiatry, as well as other diseases like cancer and heart disease. And the promise of molecular medicine is illustrated in this slide, where we begin with a disease such as schizophrenia, autism, bipolar disorder in the realm of psychiatry. We then investigate the genomes of individuals that suffer from this disease with the hope of discovering the genetic mutations that confer risk or cause the disease. With that information, we can then create animal models that carry these same genetic mutations. And often these are mice, but they're also fruit fly models and also zebrafish models. These are common biological animal models of genetic disorders because of the ease with which we can manipulate the genome in these species. Once we have these animal models, then comes a lot of hard work of applying the knowledge of basic neurobiology that we've gained by studying the brain over the last 100 years. And if we're lucky, we can apply that insight and discover what's different about the brain that carries a mutation that causes a disease. And that's called disease pathophysiology. How does the brain function differently? And once you have an understanding of how the brain is functioning differently, you may, if you're lucky again, discover a target that is a process in the brain that has gone awry that might be amenable to a pharmacological or other manipulation. This is called target identification. And finally, drug development, developing small molecules that will interact with that target and restore normal function. And finally, the hope is that we can introduce novel therapeutics for the treatment of these really devastating diseases. Now, these are really early days in making major progress in psychiatric disorders. And the reasons we haven't made more progress are threefold, and probably again are obvious. One is psychiatric diseases are highly complex. I've already mentioned the heterogeneity of autism, but the same thing's true for schizophrenia, bipolar disorder, and others. Secondly, the brain is very complex and very heterogeneous. There are many different types of neurons in the brain, and these may respond differently to these genetic lesions that cause disease. Finally, the genetics of these psychiatric diseases are highly complex. There are very few diseases, mental illnesses caused by single gene mutations. It's a complicated mixture of many affected genes or genetic disruptions that are extremely rare and have not yet been discovered. So we still have a lot left to do. And this is true in autism as well as other psychiatric disorders. So this pie chart is supposed to represent the known genetic risk architecture of autism. As I said, we're very confident it's a genetic disease, but most of the mutations that are responsible for it are unknown at this point, although progress is extremely rapid with advances in technology to sequence genes. But having said that, we now do know about the causes of autism in 15% or so of cases. And some of these are rare single gene mutations. There we go, rare single gene mutations. Other are larger segments that contain many genes that are duplicated or deleted. But we're beginning to make some real progress in understanding the genetic lesions that can cause autism. And one of the ones that I wanted to talk to you today about is this disease called Fragile X syndrome. So I mentioned Fragile X syndrome is the leading known inherited cause of autism and intellectual disability. And Fragile X syndrome is caused by the silencing of a single gene called FMR1, and the loss of the product of that gene, which is a protein called FMRP. And the advantage that this disease has given us is that we can study genetically validated animal models, and we've made great progress in understanding the pathophysiology and have identified some therapeutic targets. So just to quickly give you a quick thumbnail sketch of the history of Fragile X, it was originally recognized as a syndrome back in 1943 by very astute clinicians, Martin and Bell, that recognized that some children had the same constellation of symptoms and may have a disease that is caused by the same thing. And that thing was then discovered in the 90s, work done at Emory University by Steve Warren and also in collaboration with David Nelson at Baylor and Ben Oostra in the Netherlands. These investigators discovered the gene that was disrupted in Fragile X. And as I mentioned, it's one gene called FMR1, and it is silenced in individuals with Fragile X syndrome. And as a consequence of that, the protein product, FMRP, is not made. Shortly after the discovery of the gene, the group in the Netherlands generated a mouse that carried the same mutation. So these are mice in which this protein FMRP is not produced. So these are what we call Fragile X mice, or FMR1 knockout mice. These animals have been available now for a few years, and many investigators have taken up the challenge to study them for various reasons, as I'll show you in a second. And I'd say the breakthrough discovery that dates back to the early 2000s was a discovery that a neurotransmitter receptor in the brain called mGluR5, I'll tell you a little bit more about that in a second too, was excessively active in the brains of mice with Fragile X, and fruit flies with Fragile X, and zebrafish with Fragile X, and presumably in humans with Fragile X. And this discovery identified mGluR5 as a potential target. It's a neurotransmitter receptor. It's too active in the brain of individuals with Fragile X, and maybe we can correct the disease by dampening down signaling at the mGluR5 receptor. And so we and many other labs around the world tested this idea, and we discovered that indeed in all these animal models we could restore normal function by suppressing by about 50% the activity at this neurotransmitter receptor. And by good fortune, pharmaceutical companies, large and small, have been developing small molecules to interfere with the functions at this receptor. And these molecules are now in human clinical trials with very promising results. So, of course, there's actually a very long history to how we got to this point. And I think that what was critical here was the confluence of the information from the study of the genetics of the disease and the study of basic neurobiology. That is, how does the brain normally work, which then teed us up to understand how it might work in the absence of this protein. And we don't have time to go through all the milestones, and of course there are many that aren't listed on this slide, but the key milestone on the genetics side was the discovery of the gene, as I mentioned, by Steve Warren and David Nelson and Ben Oostra, and the beginnings of the understanding of the biology of the protein that was missing, and the generation of the Fragile X knockout mouse. These were very important discoveries. On the other side, the basic neurobiology has, in a sense, a much more tortuous path to where we stand today. And it really began, at least in my opinion, with seminal studies done in the early 1960s by researchers that were interested not in Fragile X or any human disease, but in how the visual part of the brain normally develops under the influence of experience. And what they discovered is that if you degrade sensory experience in an infant monkey, cat, mouse, or human, that you'll disable or you'll disrupt the normal development of connections in the visual part of the brain, and that will result in blindness. And so this has been a paradigm that we've studied for many years to understand the role of experience in brain development. And since one of the key features of this was the loss of synaptic strength, that is a disconnection of the inputs from the eye to the brain, there was interest in developing an understanding of the mechanisms by which synapses get weaker in the brain. And so a paradigm was developed. This LTD paradigm later was found to be disrupted in the Fragile X brain, and this is what led to the current excitement in Fragile X. So since, as I told you, I'm a basic neurobiologist, I'm going to walk you through a little bit of the basic neurobiology. So as I mentioned, it began, in my view, with the work of Hubel and Wiesel. Probably many of you learned about this stuff in school. It's been around for a long time, since the early 1960s. And as I mentioned, they are studying the development of connections from eye to brain and the role of experience. And here are the connections from eye to brain. And the important point is that they degraded vision in one eye to reproduce a human condition called amblyopia, in which an individual is perhaps born with a cataract in one eye or have a different refractive correction in the two eyes. Then there's a disruption in image formation in the eyes, and that disrupts the connections in the brain, so that the inputs that are deprived of high-quality vision will undergo a synaptic weakening. That is, the synapses that carry this information will get weaker. And there's a compensatory increase in the strength of the inputs from the other eye. So the action here is at these synapses that bring information from eye to brain. And these synapses, like 80% of the synapses in your brain, use glutamate as a neurotransmitter. So glutamate is the major excitatory neurotransmitter in the brain. Without it, your brain doesn't work at all. And so what we're trying to understand then is a modification of this synaptic transmission that's mediated by glutamate. So glutamate, the neurotransmitter, is released in response to electrical impulses that arrive along these wires that come into this part of the brain. The glutamate's released, it diffuses across a narrow cleft, and it activates receptors on the other side of the synapse, the so-called postsynaptic side of the synapse. And these receptors are those that mediate synaptic transmission. I'll give you a quick primer on glutamate receptors. Everybody should know about glutamate receptors, in my opinion. So there are really two types of glutamate receptor. Probably the most important glutamate receptor by far is this one here, this blue receptor called an AMPA receptor. And so when glutamate binds to this receptor, a channel opens and allows electrically charged atoms to flow into the postsynaptic cell, and it causes electrical excitation of the postsynaptic cell. And that's how our synaptic transmission works in the brain. If we block these AMPA receptors with a drug, it's like getting rid of all the glutamate. Your brain stops functioning. It would quickly cause death. These other two receptors, called NMDA receptors and the mGlu-R I've already introduced, have more of an ancillary role at the synapse, and their role appears to be to modify transmission through the AMPA receptor. So the modification of transmission through AMPA receptors really is the physical basis of how information is stored in the brain, both during learning and memory and also during brain development. So the star of the show today is this one here, the green receptor, the metabotropic glutamate receptor. Okay, so that's a mouthful, metabotropic glutamate receptor. It's not an ion channel. It doesn't pass electrically charged atoms. Rather, it's what's called a G-protein coupled receptor, and it activates biochemical processes inside the cell. And in our brains, in our nervous system, we have 8 different flavors of metabotropic glutamate receptor, numbered mGlu-R1 through mGlu-R8. And they are divided into 3 functionally related categories, group 1 and group 2 and group 3. And when I'm talking about mGlu-Rs, I'm talking about group 1. And the one I'm really interested in is this receptor here called mGlu-R5, metabotropic glutamate receptor 5. And we have some tools to study it, quite a few tools nowadays. One is we can apply drugs that will activate the receptor, and we can apply drugs that will inhibit the receptor. And we can use those drugs to try to interrogate the role of this receptor in normal synaptic transmission in the brain. Now, we had an idea that really dates back to the late 80s that a way to make synapses weaker was to activate this neurotransmitter receptor, the mGlu-R5 receptor. And that was the idea. 10 years later, we finally had the technology and tools to test the idea. And this is an example of an experiment that was done in my lab by a very talented student of mine, Kim Huber, back in 2000. It's a long time ago now. And what this graph shows is that this is the strength of synaptic transmission on this axis versus time in the experiment. And she has applied very briefly a drug that activates metabotropic glutamate receptors. And what you can see is that the synaptic strength goes from this value at 100%, and then it quickly depresses and it stabilizes at a depressed level. Okay, so just by applying this drug for 5 minutes, it's made the synapse about 50% weaker. Okay? So this is a biochemical trigger of making synapses weaker, synaptic depression. And it's a phenomenon that we call long-term depression, LTD. It's a form of synaptic plasticity. Now, there are many forms of LTD in the brain, but this one that's triggered by metabotropic receptors is special in one way. And the way that it's special is that it requires the immediate synthesis of new protein at synapses. And this is demonstrated by an experiment in which a protein synthesis inhibitor is introduced into one of these neurons. And in that case, this LTD response, although it's initiated, it crawls back towards the baseline. So there's an absolute requirement for new protein synthesis. Now, it had been known, thanks to really pioneering work with electron microscopy, that the machinery, the biochemical machinery for protein synthesis exists at synapses. But it was really a mystery what the role was. And what Kim's results showed is that one of those roles is changing synaptic strength, and in particular, making synapses weaker, which is an unexpected observation. So this, we sort of went on to study this the best we could. This is a model for what we think's going on. We think when we activate this metabotropic receptor, these AMPA receptors, which you remember are the workhorses of synaptic transmission, are internalized as a consequence of activating this receptor. But if protein synthesis is inhibited, these receptors come back onto the membrane and synaptic transmission is restored. But if protein synthesis is triggered by activation of this receptor, somehow this is stabilized. Okay, this is pure basic science, just curiosity-driven science. We're like, wow, what is the role of protein synthesis? So this is where our work bumped into Fragile X. And it did so because one protein that was known to be synthesized in response to activating this metabotropic receptor is this protein, FMRP. And you remember, that's the protein that's missing in Fragile X. So we barely knew what the disease was at this time. We knew there was a disease called Fragile X. We knew it was bad, but we were more interested in the protein than the disease. And we had a hypothesis, and the hypothesis was that maybe this FMRP is a critical regulator of this long-term depression response. Okay? So that was a hypothesis, and to test the hypothesis, we got the animal from Steve Warren that was the Fragile X mouse that didn't have any FMRP to study this LTD response. Well, to make a long story short, what the results showed is that instead of the FMRP being required for this LTD, the FMRP actually is a negative regulator, okay? And this converged with the biology of the molecule that was then emerging, which is that this protein, FMRP, binds messenger RNA at the synapse, and it prevents protein synthesis. So FMRP, instead of being one of the key products of protein synthesis, is actually a key regulator of protein synthesis at the synapse. Okay, so we have a balancing act between activating protein synthesis through mGluR5 and inhibiting protein synthesis through FMRP. And the discovery that led to this model was Kim's discovery that this long-term depression readout that we were studying, instead of it being impaired in the Fragile X knockout, it was exaggerated in the Fragile X knockout. Okay, so here's her data showing now in the yellow symbols, this is the LTD response in the Fragile X knockout mice, showing that instead of a reduction in the LTD response, we got more of the LTD response. Okay, so that was pretty exciting. We realized that FMRP might be a very important regulator of protein synthesis that's stimulated by this receptor, mGluR5. Well, we broadened the lens at this point, and we asked ourselves, well, what other functions in the brain have been attributed to mGluR5 stimulated protein synthesis? And it turns out that there was a growing list. This is back in the early 2002 to 2001. There was a growing list of functions that have been ascribed to mGluR-dependent protein synthesis. And we just asked ourselves the question, what if all these functions were exaggerated in Fragile X? How many of the symptoms of the disease could we account for? And the startling answer was a lot. We thought we could account for a lot of the symptoms of Fragile X syndrome. In other words, we had discovered a thread that could connect syndromes as widely varied as intellectual disability, altered gastrointestinal function, and seizures, okay, by this one mechanism. So we threw the idea out there to our colleagues, and we called it the mGluR theory of Fragile X, which I'm sure is a gross oversimplification, but it's a way to organize thinking and generate experimental tests. And so the idea was that the psychiatric and neurological aspects of Fragile X are a consequence of exaggerated responses to activating mGluR5 in particular, based on the assumption that excessive protein synthesis downstream of this receptor is pathogenic in Fragile X, is what causes the symptoms of the disease. And there was a very important prediction. And the prediction was that inhibiting this receptor could restore a normal balance in the brain and restore normal function, okay? And so we and other labs set out to test this idea, this prediction. And the way that we did it was we crossed the Fragile X mice that harbor the Fragile X mutation with another line of mice that we had engineered to have 50% the normal level of expression of this mGluR5 receptor, okay? Now, reducing mGluR5 receptors by 50% has very, very mild consequences on its own, but it was sufficient to completely correct the symptoms of Fragile X in the mice. And this was the paper that Gould-Dolan published for my lab some years ago. So this was an exciting genetic test or proof of principle that this could be a therapeutic target. In the meantime, however, researchers all over the world were using the pharmacological tools that were available, a small molecule that we call IMPEP, which is an inhibitor of mGluR5, and they found that in many different assays of altered function in Fragile X, IMPEP could ameliorate or correct those disturbances. And so I just have copied the title pages of some of these papers, and I want to point out that this is not just in mice, but also very importantly in the fruit fly model of Fragile X and also in the zebrafish model of Fragile X. So, you might be surprised to know that we can model the disease in a fruit fly, but we certainly can, because there's a very similar gene in the brain of the fruit fly, expresses a very similar protein that has a similar function. We can knock that out, and amazingly, that even in the fruit fly, distantly related species, these mGluR5 antagonists had beneficial effects. So what this suggests is that there's an evolutionarily conserved relationship between the mGluR5 receptor and the Fragile X protein. And that gives us great confidence that the same thing should work in humans. So just to sort of recap what I've told you so far, is that there exists in your brain a molecular machine that ensures that proteins at the synapse keep up with demand. And demand is registered in the brain by the release of glutamate and the activation of this glutamate receptor, this mGluR receptor. So the more active the brain is, the more mGluR receptors are activated, and this stimulates protein synthesis, that is the translation of messenger RNA into protein. Now, like any machine in the body, there are important regulatory mechanisms, and the FMRP protein is one of those. And the FMRP protein normally serves as a brake on protein synthesis, okay, so it doesn't run out of control. Now, we've known for some years that synaptic performance in the brain requires an optimal level of synaptic protein synthesis. We know that there are a lot of diseases that are actually characterized by impairments in synaptic protein synthesis. So what we believe is that the function that relates synaptic performance to protein synthesis is like a bell-shaped curve, an inverted U-shaped curve. And so that there's a minimal level is required for optimal synaptic performance, but if you have too much, then there's an impairment. And that this is one of the regulatory pathways that controls this. In Fragile X, what happens is you remove the brakes. It's like taking your foot off the brakes, cutting the brakes off the car. All you have is an accelerator. And this leads to exaggerated protein synthesis and impaired function. And a strategy for correcting this is to take your foot off the gas, okay? Inhibit signaling through the mGluR5 receptor, pushes that ball back up to the peak here, which is the optimal synaptic function. So I think it's really exciting that the idea behind the experiments behind this idea. We're in the early 2000, 2001, and now in 2011, we're already beyond phase 2 clinical trials and into phase 3 clinical trials to test this idea in humans and hopefully introduce a therapeutic that will help individuals affected by Fragile X. And the compounds in this pathway that are in phase 2-3 clinical trials are, first of all, a number of different molecules that are specifically targeting the mGluR5 receptor, and another approach that is targeting another receptor that indirectly inhibits mGluR5 by inhibiting the release of glutamate in the brain. Okay? And so these trials are sponsored by Seaside Therapeutics, which is a small biotech company in Cambridge that I co-founded, and the pharmaceutical giants Novartis and Roche also have trials in Fragile X underway. And I'll tell you just a little bit about the Seaside trials since I know something about them. The drug that they are advancing is called baclofen. Technically, it's actually one of the stereoisomers of baclofen called R-baclofen. It's a drug that activates a receptor in the brain called a GABA-B receptor, and as I mentioned, GABA-B receptors normally suppress glutamate release in addition to other actions. The reason baclofen was an attractive path forward is that baclofen is already in clinical use for the treatment of intractable gastroesophageal reflux and also in cerebral palsy. So there's a lot of clinical experience with baclofen, and so we decided we would take this forward to see if we could treat Fragile X and autism. And this is the design of the phase 2 study that's been completed. So individuals with Fragile X were brought in, they were screened, they were divided into 2 groups, and group 1 received the drug. The drug was titrated up, and they stayed on the drug for a total of 4 weeks, and then it was titrated down. The other group received placebo during the same time period, and of course the subjects and the clinicians were blind to whether they were receiving drug or not. And then there was a crossover so that those that had been treated with drug in the first phase of the study were treated with placebo in the second phase, and conversely, those with placebo were treated with drug in the second phase. And this is a great way to use the same subjects as their own control, and also that every subject will receive the drug. And then there were follow-up assessments. And a lot of things were measured in this study. And it was a phase 2 study, so this is really considered exploratory and not really conclusive. But we were extremely excited by the finding that there was an improvement, particularly in the individuals that were characterized by lower sociability, which you remember is one of the key characteristics of autism. And so this is what's called a clinical global impression. That is, a clinician just says, they just rate the patient, they're blind to the treatment, is the patient better or worse, and how much better. And so those that treated with placebo, you see a big spike here at no change, not surprisingly. But those that were treated with the R-baclofen, called STX-209, showed a shift in this distribution to a peak at much improved, showing a positive response. So based on these data and others, Seaside is continuing forward into phase 3, both in Fragile X and in autism, with sociability, social withdrawal as a clinical endpoint. Okay, so this is the same diagram I showed you a few minutes ago. This is just a simple cartoon to represent the molecular machine that regulates protein synthesis at the synapse. And of course, I'm sure you're aware that this is a gross oversimplification, and I can make it a little bit more complicated but without capturing the full complexity. And that's what this diagram is. This is just showing some more of the cogs in the machine where we have, here's a glutamate, here's the receptor, and down here is protein synthesis. And we have a number of other proteins and gene products that are likely involved in the regulation of this process. And I think what's tantalizing about this is that many of these same proteins or genes are affected in other disorders that lead to autism. Okay? So that's what's representing these different colors. I can't remember the color code, but some of them are, right, okay, so these are rare single gene disorders So a single gene is affected, and one of the consequences is autistic behavior. That's the elements in red. The elements in green, what color is that? I'm colorblind. Are again, extremely rare mutations that cause non-syndromic autism, that is almost pure autism. These are proteins that are up here. And finally, these are genes that show an increased proportion of rare, potentially structurally deleterious mutations in autistic patients. So it leads us to believe that maybe we have stumbled upon a pathway, a biochemical pathway, that is altered in the autistic brain. So what would be wonderful is if many of these genetic lesions converge onto a limited number of pathophysiological mechanisms. As I said, the genetic heterogeneity is large. There are many genetic mutations that lead to autism, hundreds. But the problem gets a lot simpler if they all have as a common consequence a disruption of a biochemical process, particularly a biochemical process that we have the hopes of targeting with a drug therapy, such as we are able to do in Fragile X. So the question in the field right now, the burning question, is that will treatments developed for one cause of autism spectrum disorder like Fragile X be effective for others? So just to wrap up, Fragile X is poised now to be the first neurobehavioral disorder where treatment has been developed from the bottom up, from gene discovery to pathophysiology in animals to human clinical trials. And this represents the convergence and the confluence of progress in genetics and also basic neurobiology. Genetically engineered animal models have been absolutely critical for this step. And as I think is clear, there's a tremendous amount left to be done. And finally, I just want to thank those who have funded our work. We've received funding from a lot of different sources. Our bread and butter has been from the National Institutes of Health. And it's interesting to note that we've gotten money from a lot of the different institutes that didn't know that ultimately they were funding projects that might lead to a cure for autism, including the National Eye Institute, the National Institute for Neurological Disorders and Stroke, the National Institute for Mental Health, and of course the National Institute for Child Health and Development. And we've also received funding from private foundations, most notably the Howard Hughes Medical Institute, the Simons Foundation, which has an initiative in autism, and also FRAXA, which is a small but extremely effective nonprofit organization that has funded work in Fragile X research. And with that, thank you very much.
Maybe a simple answer or not. The treatments that they're looking at for Fragile X right now, if patient X is taking it and it's working for them, and they stop taking it, is it like schizophrenic patients who go off their meds, will they revert back to a certain stage?
That's a good question. And we don't know the answer at this point because, I think you can appreciate that the best hope for lasting treatment would be to start early. And I think that there still is the hope that timely treatment during early brain development, early postnatal brain development, may be sufficient to have a lasting benefit even in the absence of continuous treatment. But as it stands now, most of our information is based on treatment in adults or young adults where discontinuation of treatment will cause them to revert. So for instance, in the trial that I told you about with the arbaclofen, they definitely regress once they go off the treatment. And the FDA has granted Seaside the right or the opportunity to allow the patients that were in that study to continue on drug if they saw a benefit. And the vast majority of those patients have elected to stay on the drug. So.
I'm curious about the behavioral health aspect of Fragile X. I mean, I know a lot of children with autism have extreme sort of emotional issues and they lash out because they aren't able to speak what they actually feel. And I'm wondering whether you have created sort of coursework for when you're doing this medication, is there also sort of a behavior modification that goes along with it?
Yeah, you know, that's a great point, and that is something that we intend to incorporate in the future clinical trials of comparing not only drug treatment alone, but drug treatment in combination with more intensive behavioral therapy as well. Because a lot of the treatments that we're talking about are sort of plasticity promoting in the brain. And so, in a sense, the treatment alone may not have the effect, but that may greatly amplify the effect of behavioral therapy. So it's certainly on the radar.
I just had a question about the design of your clinical trial. Did the patients know that this sort of infinity-shaped crossover thing was happening? You know, where they...
Yes.
Yeah.
Yeah. They knew. They knew it was a two-arm clinical trial, and that during one arm they'd be on active compound and one arm they'd be on placebo.
Do you think that could have had any impact? I mean, like somebody goes, well, I was better and so now I'm going to be on the placebo, or there was no difference, so now I'm going to,
You know, the thing that we have to, the challenge is to beat the placebo effect in a lot of these trials. For instance, so we saw a good effect in what I just showed you, the clinical global impression. We saw a lot of good effects in other measures. But one of the measures we expected to see a good effect in was an irritability subscale. And we saw a good effect, but it wasn't better than placebo. And within the Fragile X community now, there's a great awareness and anticipation that these treatments are in the works. And that, in a sense, it's worked against us a little bit because I think a lot of the parents are anticipating the drug will work. So this actually probably leads to a more exaggerated placebo effect than we would like to have a successful clinical trial.
It's already existing in our baclofen. How does Seaside continue the need to want to do the research? Is it because they can get a patent for the new indication, or how does that work for a treatment that's been around for a long time?
Okay, so there are two things. One is that patent for a new indication, also formulation, and finally, this is the R enantiomer of baclofen, where only racemic baclofen has been in clinical practice before. So those are the strategies used to protect the investment in baclofen. Yeah.
So for a single gene condition like Fragile X, I can see where it would be beneficial because you can identify early. I guess I'm wondering, with many autistic children, they're not being identified until they're older, maybe 2-3 years of age, and so while your trials are showing good results right now for young adults and adults, how would you be able to identify for non-Fragile X autistic individuals, how would you be able to identify if this would be in the correct pathway? Or?
Well, you know, historically, those with Fragile X were not diagnosed at birth. They were actually diagnosed after missed developmental milestones, and then that was one of the tests that was then administered to distinguish whether it was Fragile X or autism of unknown cause thing. So, of course, newborn screening would be the best. But sort of the other part of your question, which is how are we going to know if someone that doesn't have the Fragile X diagnosis is going to respond to this treatment, particularly given the heterogeneity of not only the disease, how the disease presents, but what causes the disease. And that is a very serious problem. And the path forward is going to be to compare the pathophysiology of Fragile X with other single gene disorders that cause autism, and ask whether a treatment developed for Fragile X will be similarly beneficial in these other disorders. And it's a critical question, and it's really what devils the pharmaceutical industry in the treatment of all mental illnesses, which is that probably all illnesses, but there are responders and there are non-responders, and there are some people that are made worse by a medication. And that, needless to say, makes clinical trials very costly, and that's huge numbers. And what we'd all like to know is, can we predict a treatment response a priori? In Fragile X, we have that advantage that we can measure the gene. We can say, this individual has Fragile X. they will respond to an mGlu-R antagonist or baclofen. But without that kind of knowledge, we'll be shooting in the dark. So there's a big effort underway right now trying to understand so-called biomarkers that will enable us to predict treatment response in these other populations.
You mentioned how this is kind of a milestone in terms of building up a treatment from the basic neurobio to genetics. And I'm wondering, from your perspective in the basic neurobio, are we going to see a lot more of this over the next few years or the next decade? Are there going to be a lot more treatments? Can you talk about that?
Short answer is yes. You know, I highlighted the work in Fragile X, but there's also been some really nice work in another disease called tuberous sclerosis, another called neurofibromatosis, another called P10 hamartoma syndrome. These are all very rare diseases that affect children, but we've gained some insight into the underlying pathophysiology, again, because we've been able to study the animal models of the disease. So it really is, there's going to be a revolution. I mean, this revolution has been predicted for a long time, right? But I think we're right now on the cusp of realizing that promise. Now, in some ways, we're lucky with the single gene disorders because we can knock them out in mice and so on. But some of these disorders are going to have much more complicated genetics than a single gene, and that's going to make matters more difficult for modeling them. And so a very exciting prospect that's underway now is to use what's called induced pluripotent stem cells that are derived from the cells of the patient. That is to say, cells from the patient are harvested and actually converted into neurons, and then we can ask how those particular neurons function differently in that patient without necessarily knowing the full complexity of the genetics. And so that's a really exciting development as well.
A portion of the autism community has a lot of biological theories about how one thing or another can cause autism. Do you know whether any of those biologically based theories have anything to do with glutamate receptors and so forth?
No, I don't. I mean, you're right. There are a lot of ideas out there for what triggers autism. I mean, I think we know. I think the argument that the genetics are of critical importance is beyond reproach at this point. However, when I said you had a 90% chance of developing autism if your identical twin has autism, that's not a 100% chance. So there is a role for environmental effects, and we don't fully understand them. And so it's going to be, but you know, as someone who spent most of his life trying to understand how experience modifies synaptic transmission, we know a lot about that process too. So it won't be hard to fit these two things together. It's going to be, you know, gene, time, experience interaction that's going to screw things up, and hopefully we can make them better.
Thank you very much.